بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. I'm Dr. Muthusen, GKMC at Swagger. Today, we will discuss the development of the eye, how the eye is formed, and how see this eye at the beginning. So we will start it from the very beginning. So I'll try to remind you the general reality, the very initial stages. I will say about the third week. So there were important events of the third week, in which the first one was, and the important one was the introduction of a third germinal layer, which is called the intra-embryonic mesoderm. The other was of the formation of the neural tube, along with other events. So we will concentrate today, and we will start from the formation of the neural tube. If you remember this diagram, this was the very beginning of about the third week, that the surface ectoderm, when it grows, it becomes thickened, and it is responsible for the formation of the nervous system at the end. So this is called neuroectoderm. Previously, it was called ectoderm, and then when the cell grows by mitosis and it becomes multi-layered and become thickened, so it is called a thickened the neuroectoderm. With further passage of time and the development, and increase in the growth, it tries to accommodate itself in a limited space. So indentation starts in this, and you can see the indentation here. It was a flat neuroectoderm. Now here indentation is appeared in the bed, and this is increased, and it takes the form of a groove at the end. These ends will come up. Similarly, this end will come up. Finally, they will close to each other, and it will be converted into a neural tube. Now it is a groove, then it will be converted. These are the neural crystals, different colors. They will dissociate in this place, and they will migrate to various other spaces. We have discussed that in details. This is the surface ectoderm, and finally, this surface ectoderm is separated from this neural tube. When the neural tube is formed, you can see here in this place. When the neural tube is formed, this surface ectoderm is separated from it. So it is about at the age of 21, 22nd days, the neural tube in the 4 or 13, 14 somatid state. We can see this picture. The previous picture was this one, the lower one. This is about 22 days, and this is actually the proximal part or the cephalic part of the neural tube, which is responsible for the formation of brain, forebrain, midbrain, and so this is an area of the forebrain. With further growth, you can see here a little bit differentiation here. This is forebrain, and these are called the vesicle, or we see at the lens space hood in the beginning, and the inside they are forming vesicle. These vesicles are communicating with the forebrain, and with the passage of time, the lumen becomes narrow, and it is converted into an optic stalk. You will further appreciate in the next picture. I will tell you. Here you can see that portion of the optic vesicle, which is the part of the forebrain, and the lens with the surface ectoderm, which is appeared green in color. When they are growing, they coming towards each other. And now they have a gap in between, but with the passage of time, this gap is reduced, and finally they come very close to each other and they lie in a position with each other. When this surface flow down, such as this area, and at this level, there is a induction that reduces it to thicken. This surface will become further thickened, and later on there's a formation of a concavity in this forebrain area, like just like a Bowman capsule. If you, I didn't told you, we will discuss it in detail. It's just like a Bowman capsule. A cup, and a cup is formed, and the surface of the dome, like a vesicle, goes inside, and they come in contact. This surface ectoderm will come that it become like a groove, and finally it will make a tube or lens-like structure, and this will be separated from this surface ectoderm and will be responsible for the formation of the lens of the eye. So the lens is basically ectodermal in origin. It has been derived from the surface ectoderm and has detached from it. In next picture, you can see the previous one as well. This is Dancephalon forebrain, and these are the small vesicle, and with the passage of time they have growth, and the surface ectoderm has come in contact with this. Previously, the surface ectoderm was outside, and now it is in contact with this. After this, this has become concave, and this become a double layer. One is the outer layer, this one. And this one, when it goes further in, it will become the inner layer like this. And this will make the lens, which is actually the derivation of the surface ectoderm. So the lens is the part of the surface ectoderm, while the optic cup in the form of double layer, the outer layer and the inner layer, it is actually from the brain, it is called optic stalk. If we take the optic cup and optic stalk, and this diagram you can see there, and the line of interrupted line shows the line of section. If we cut this structure at this level, we will see the structures A, B, the structure B. If we cut the structure A at this level, at an interrupted line, we will get, and we see it transverse section. So we will see like this. You can see here that the outer layer, there is an inner layer, and there is a gap in between the two, and this structure will form the retina at the end. This optic, actually it is optic stalk now, but later on it will be converted into the outer layer and the inner layer in the space in between the two. This space is called intraretinal space. If you see here, during this development, there is a gap at the inferior surface of this optic stalk. This is called choroid. This is called choroid fissure. This is a gap, and with the passage of time, this gap will close, and this fissure will be converted into a complete tube-like structure, which is called optic canal. And this carries, you can see in the very center, a small dart-like structure. This is the transverse section of the artery which supplies the lens. And this is called the hyaloid artery. And the intraretinal life, it supplies the lens and the ciliary bodies. And with the passage of time, this then close at about 7 to 8 weeks, it then close and it become converted into a complete tube stack, complete tube-like structure of optic canal. The lower down are the electronic micrograph of this picture. If you see by electronic micrograph, you can see these things like this. Here you can see this development of the eye started about the beginning of the 4th week, you can say 21, 22nd days, say, it starts. And it develops. At 7-week stage, you will see like this. It is a 7-week embryo, just 7 weeks. That is less than 2 months. It is almost like lichen at an eye. If you concentrate on this area, this is the optic stalk. I told you that it has got the outer layer and data. So I'll tell you again, this one. This is the outer layer, this one. This one is the inner layer. These are the outer and inner layer of the optic stalk, and finally it will develop into a retina. So see here now, this is 7-week embryo. It is almost completely identifiable that it's a nice structure. So you can see this optic stalk like a rounded circle. The posterior 4/5 of this, like this. It is divided into two: the outer pigmented layer of the retina, the outer pigmented layer of the retina, and then the inner neural layer of the retina. And both these layers, the pigmented layer and the neural layer, they are separated from each other by a space which is called the intraretinal space in the beginning. Later on, with the development, this space obliterates and both these layers, that's pigmented layer and the neural layer, they fuse and they become one. And here you see that they are concentrating on the optic nerve. This is the highlight vessel, which I told you that it goes to the choroid fissure, and this highlight artery supplies this lens and the ciliary body area. Here you can see that development is going on, and it is a stage of about 15 weeks. 15 weeks pregnancy, hardly of 4 months, you can say, or less than 4 months, 3.5 months. Further development has been taken place, and you can see the development of the interior 1/5 of the retina as well. The posterior 4/5 was like this, that the outer layer was pigmented layer, the inner layer was neural layer. And this neural layer then converts into a multiple layer that's called the photosensitive receptor, that is rods and cones, and the outer nuclear layer, the inner nuclear layer, and the ganglionic cells, and finally they are converged into the fibers of the optic nerve, and which is connected to the brain. The interior 1/5, you can appreciate here that it is converted into a ciliary muscles and ciliary bodies, and an irido-pupillary surface. So we can say that at the end of the 6 weeks, we can see like this, that the interior differentiation, you see these structures, these are the mesenchymal tissue, all these. These are the mesenchymal tissue, and they are surrounding this primordial eyeball. And the interior portion of this will be responsible for the production of the ciliary body and the ciliary muscles. This is the details of the retinal layer. I told you that the outer pigmented layer, then again, which is called the intraretinal space, and then starts the inner layer, which is again divided or further differentiated into the photosensitive receptor, that is rods and cones. Then the outer, these are the outer nuclear membrane outer nuclear layer of the rods and cones, and then the inner nuclear layer are called the bipolar cells as well. These are the ganglionic cells. And finally, they converge on the fibers of the optic nerve, and they are converted, they are sent to the brain. These are the iris and ciliary body. How they develop? I told you that these tissues are loose mesenchymal tissues. These tissues are loose mesenchymal tissue, and here you can see, appreciate the development of the fibers, which is called the ciliary muscles. These are the ciliary muscles. They are developed in the surrounding of the eyeball in the loose mesenchymal tissue, and these are the ciliary processes. These ciliary processes are responsible for the production of the aqueous humor in the eye, which circulates and gives nourishment to the avascular cornea and ciliary bodies. Similarly, the muscle, intrinsic muscle, that is called sphincter pupillae and the dilated pupillae, they are developed in the loose mesenchymal tissue very near to the inner unpigmented layer of the iris. And these are responsible for the change in the size of the pupil. These are called the dilated pupillae, which will dilate the pupil, and sphincter pupillae, it will distend. So these muscles are actually loose mesenchymal tissue outside the eyeball, and they are responsible for the size of the pupil. You can see the whole normal structure of the eye in this. This, you say, this is the retina, this is the optic nerve. This is a lens which is actually ectodermal in origin, it comes from the surface ectoderm. These are the interior 1/5 of the retina, this iris and this ciliary bodies like this. In front of that is the aqueous humor. This is the cornea. This is almost the final shape. And this is the outermost is the sclera. And this is in continuation posteriorly with the dura of the optic nerve. So, in a nutshell, you can say that there is mesenchymal tissues and compact surface ectoderm and converted into and optic vesicles. Then the surface ectoderm gave rise to the lens placehold, and this optic vesicle will also will give rise to the optic cup, then optic stalk, and then the formation of the retina, the pigmented layer, and the neural. So, optic vesicle will convert into optic cup and optic stalk, and then they will be responsible for the formation of the retina. The surface ectoderm will give rise to the lens placehold, then lens vesicle, and then finally the lens. And similarly, the surface ectoderm also give rise to the cornea. Now we'll talk about the chambers of the eye. That is the interior chamber, posterior chamber. Interior chamber is a space. Boundaries are, it is between the iris. Interiorly it is bounded by the iris, and posteriorly by the lens and the ciliary bodies. This space is filled with aqueous humor, represented by AH. As I told you that this aqueous humor is formed by the ciliary processes of the ciliary body. And this fluid is communicating with the posterior chamber, but by the pupil. Pupil is a space in between the anterior chamber and posterior chamber, and this is a clear fluid and is responsible for circulation and will give a nourishment to the avascular cornea and lens. Echosimmer provide nourishment to lens and cornea, as I told you. It is produced by the ciliary processes of the ciliary body. So naturally, it will be drained somewhere as well. So it is drained through venous sinus of sclera, which is called canal of Schlemm. So if there is a disturbance, usually it is reabsorbed into the bloodstream or venous system. If there is a blockage of the canal of Schlemm, which is the main drainage for this, so the production is going on by the ciliary processes and it will be accumulating here. So it is responsible. There is no drainage and it will be accumulating. So it is responsible for the increased intraocular pressure. And this is also called the glaucoma. Increased intraocular pressure is called glaucoma. So the blockage of the canal of Schlemm will be can be one cause of the glaucoma as well. This was the normal development that how they developed. Now, if there are some abnormalities occur, so these are number coloboma. What is coloboma? I told you that the optic stalk has got the choroid fissure, and this choroid fissure has been converted into stalk. But if it fails to close, it results in coloboma. The other is congenital cataract. If in the German measles affect the mother in about 4 to 7 weeks of development, so there will be abnormality which will cause the congenital cataract. Macrophthalmia, the very small eye or the less development eye. Then the other is the anophthalmia. Anophthalmia is again there is no eye or the absence of the development of the eye, and it is associated with other cranial abnormalities as well. Coloboma, when the choroid fissure is failed to close. Congenital cataract, when female is infected with German measles, specifically at the 4 to 7 week of pregnancy. Aphakia and aniridia, loss of iris. There is no iris in that. Loss of development of the iris is called iridia, and the other is iridia, and the other is cyclopia or synophthalmia. Cyclopia usually called single eye, or synophthalmia is called the fusion of both the eyes at the center. Almost both are the same. Either we call it the single eye or the fusion of both eye in the center. Usually the cause for this is the single eye or fused eye is the loss of the midline tissues during any insult to the tissue at the very early stage of the pregnancy, that is in the first 4 to 6 weeks, and it is always associated with defective cerebral hemisphere development as well. So it was all about the development of the eye. Thank you very much for the office.